Welcome to The Explainer. Today, we're unpacking an absolute biological masterpiece, the Phototransduction Cascade. This is the exact mechanism that takes a simple, tiny particle of light and translates it into the vibrant, rich visual world you're experiencing literally right now. I mean, think about this for a second. How does a single, solitary photon, the smallest possible packet of light energy, actually create vision? It's wild. The human eye is so extraordinarily sensitive that just one tiny photon striking the retina can trigger a measurable biological response. It's kind of mind-bending to think about the sheer scale of it. So today, we're going to look at the exact microscopic machinery that makes this incredible feat possible. Okay, let's dive into this. We'll start with the magic of vision, then look at catching the light, trace the molecular domino effect, see how the signal amplifies and resets, and finally, look at what happens when the cascade fails. Section 1. The magic of vision, zooming into the photoreceptors. To really appreciate this process, we need to completely shift our scale. Let's zoom all the way down into the outer segments of our photoreceptors. Deep within these rods are dense stacks of discs packed with rhodopsin. And rhodopsin is the undeniable hero of our scotic, or dim light vision. It consists of an opsin protein tightly bound to an 11-cis retinal chromophore, which, by the way, is derived from vitamin A. Because these molecules are just so incredibly densely stacked within the rods, they absolutely maximize the chances of capturing even a single stray photon in the dark. Section 2. Catching the light, the inciting incident. Now, what's really interesting about this slide is that exact inciting fraction of a millisecond. In complete darkness, retinal sits in a bench shape called 11-cis retinal. But the instant a photon strikes, bam, it absorbs that energy and instantly snaps into a straight shape called all-trans-retinal. This shapeshift is literally a molecular switch that alters the entire opsin protein, creating an activated form called metarhodopsin 2. So phototransduction isn't just some abstract chemical change, it's a structural, physical event. To understand this switch, we really have to look at the baseline state first. In the dark, our eyes maintain a steady dark current. Basically, sodium flows in because a molecule called cyclic GMP holds the cellular doors wide open. This keeps the cell slightly depolarized and constantly releasing a neurotransmitter called glutamate. So the biological problem for the eye is, how do we disrupt this constant steady current to signal that light is actually present? Well, the solution is the trigger itself. Activated rhodopsin ultimately slams those sodium doors completely shut. And cyclic GMP, that's the ultimate key to unlocking this whole sequence. Section 3. The molecular domino effect, the G-protein cascade. Let's move to and see how this builds. Prepare yourself for a high-speed chemical reaction. Imagine microscopic dominoes falling in rapid succession. Step 1. Our newly activated rhodopsin bumps into an intracellular G-protein called transducin, waking it up by having it exchange a molecule of GDP for GTP. Step 2. Transducin races over to stimulate the enzyme phosphodiesterase, or PDE for short. Step 3. PDE acts like microscopic molecular scissors, just cutting up our all-important cyclic GMP into an inactive form. And finally, Step 4, without cyclic GMP there to hold them open, those sodium channels slam shut. The dominoes have officially fallen. Which leads us to a brilliantly counterintuitive twist. Unlike most neurons in the body, photoreceptors actually respond to stimulation by turning off. By locking out those positive sodium ions, the cell becomes hyperpolarized. It literally becomes more negative to tell the brain, hey, there's light here. This sudden decrease in glutamate release is exactly what modulates the next layers of retinal cells, like the bipolar and ganglion cells. They process contrast, spatial information, and light intensity before the signal even leaves the eye. Section 4. Amplification and the reset, powering up and powering down. And this brilliantly illustrates a crucial point. How does this tiny microscopic event become loud enough for the brain to actually hear? The answer is biological amplification. Just one activated rhodopsin molecule doesn't just activate one transducin. It activates multiple, and those activate multiple PDEs, which in turn destroy thousands of cyclic GMP molecules. So one tiny photon creates a massive cascade of shutdown sodium channels. This incredible biochemical amplification is exactly why you can easily navigate a room by the faint light of a single candle. But, a system this sensitive must hit the reset button quickly, or you'd just be blinded by a permanent afterimage. To set the dominoes back up, activated rhodopsin is quickly neutralized by arrestin proteins, while enzymes like guanylate cyclase step in to regenerate that cyclic GMP and reopen those sodium channels. Now, while the initial light strike takes mere milliseconds, the enzymatic recycling pathways require the help of the retinal pigment epithelium and can actually take minutes. This prolonged recovery time to physically reshape the retinal back to its 11-cis form is exactly why it takes your eyes quite a bit of time to fully dark adapt when you walk from bright sunlight right into a pitch-black movie theater. Section 5. When the cascade fails. Clinical realities. We've spent this explainer marveling at flawless biochemistry. But what happens when it breaks down? Well, this cascade, while incredibly powerful, is also highly delicate. When patients have genetic mutations affecting these specific phototransduction proteins, or even just a lack of the vitamin A necessary to build the retinal chromophore in the first place, the entire system fails. So the crucial point is that these disruptions lead directly to inherited retinal diseases. We're talking about conditions like retinitis pigmentosa, congenital stationary night blindness, and cone dystrophies. Disruptions in this microscopic cascade literally lead to severe, life-altering blindness. But there is some truly incredible hope on the horizon. Modern science is pioneering futuristic therapies as we speak. We're seeing things like gene therapies to correct those specific mutations, retinal prostheses, optogenetics, neuroprotection, and even artificial photoreceptor systems. But here's the major takeaway. None of these breakthrough therapies are possible without a precise, foundational understanding of the normal phototransduction physiology we just walked through. You simply have to intimately understand the masterpiece before you can ever successfully repair or replace it. Which brings us to a final provocative thought to leave you with. Can mastering this microscopic cascade lead to the ultimate cure for inherited blindness? The exact, intricate processes that allow a single photon of light to trigger your perception of the world might just hold the absolute key to restoring sight to millions. It's a brilliant testament to the profound impact that understanding basic biology can have on the future of clinical medicine. Thank you for joining me on this explainer. I really hope it illuminated this fascinating topic for you. Keep wondering, keep learning, and I'll catch you next time.